Welcome to everyone joining online for today's NABS Masterclass. My two presentations today will provide a pathologist's perspective on plant poisonings with a specific focus on plants relevant to Northern Australia. So why do poisonings happen? The outcome of an animal being poisoned is reliant on a complex interaction of multiple factors. Generally speaking, we group these into animal, plant and environment. Animal factors include species, age, degree of hunger, familiarity with the environment, and level of adaptation or previous toxin exposure. Plant factors are related to its palatability, stage of growth, or physical condition, while environmental factors may involve things such as weather, nutrient supply, or the presence of plant pathogens and pests. It's important to note that most livestock poisonings happen due to unnatural circumstances, which lead to the critical intersection of these factors. So how can we tell if a mortality event such as this is due to a poisoning? Poisoning should be considered a possibility when there is simultaneous onset of illness or death in multiple animals, or illness closely follows a change in feed, location, or weather. This photo was taken from an incident where 600 wethers were tracked for 2 days without food. They were then released into these holding yards, and within 3 hours there were 250 deaths. And this was a case of acute oxalate poisoning. So now that we might be suspicious of a poisoning, how do we go about making a diagnosis? Just like any other disease investigation, it is important to be thorough and follow a systematic approach. Firstly, obtain a thorough history from the property owner or manager. Include animal-specific information and any particular circumstantial factors that may be critical in implicating a source, such as a change in weather, new batch of feed, or recent animal movements. Referring to something like the NABS disease mapping resource can assist in prompting you through all the necessary steps. Secondly, animals should be examined. Examination of both live affected animals and recently dead animals will allow you to determine the presenting syndrome. Assigning a presenting syndrome allows you to formulate a list of differentials and guides you on what samples may be necessary to collect in order to confirm a diagnosis. Where possible, a complete range of samples should be collected from both live and dead animals, including samples specific such as ocular fluid and gastrointestinal contents. Whilst the lab may not utilise all the samples you've submitted, it is better than not having access to samples that may have been critical in reaching a definitive diagnosis. It's also vitally important to inspect the environment in which the animals have access to. Examination of supplementary feeds, pastures, and water sources is critical. A thorough environmental examination will involve a paddock walk with the property owner or manager. And remember to always look for evidence of plants that have been eaten. Now, after gathering a complete history, examining animals, collecting samples and inspecting the environment, it's very likely you will either have a tentative diagnosis or a very short differential list. This is where you may like to send samples in for laboratory confirmation. It is essential that you communicate all your findings on the laboratory submission form, including history, clinical signs, and necropsy findings where appropriate. List the samples you've collected from both animal and plant sources, and where relevant, ensure you have followed any specific preparation and handling procedures necessary. For example, pouring off serum, freezing aqueous humour, or pressing plants for identification. If you are unsure about any of these processes, please contact your lab directly. As I've just discussed, by determining which organ systems are affected and assigning your case a presenting syndrome, this allows you to narrow down your list of differentials. I have listed the presenting syndromes here in order in which I think is most common, and will discuss the top 5 syndromes, including the plants most likely to be responsible for these, in today's 2 presentations. Note those marked with an asterisk, namely sudden death, acute liver necrosis, and photosensitization are highly suggestive, particularly in ruminants, that a poisoning is involved. The other presenting signs share their etiologies with infectious, genetic, metabolic or nutritional diseases. However, there are a number of possible toxic causes seen in these other syndromes that should also be considered. So for each of the 5 presenting syndromes that I will discuss today, I've included a summary slide which will list the major causes and those that I will discuss in detail. These are in bold, with the less common causes, but those still relevant to Northern Australia, in normal font. So starting with sudden death. This is one of the most common syndromes encountered in livestock, particularly where intoxication is suspected. Animals may be simply found dead or observed to die within a few minutes to hours of becoming ill. The most common phytotoxic causes of sudden death we see in livestock include cyanide and nitrate-nitrite poisonings, acute oxalate poisoning due to soluble oxalates, a variety of myocardial phytotoxins, and poisonings by plant that produce acute liver necrosis. Now, unlike other phytotoxic causes of sudden death listed here, where gross pathology is generally unremarkable, the presenting syndrome of acute liver necrosis is fairly recognisable due to typical necropsy findings, and so this syndrome will be discussed separately. Cyanogenic glycosides, also known as cyanide or prussic acid, can be found in numerous plant sources, including grasses, forbs, shrubs and trees. The most common source of poisoning for livestock are the sorghums. Because the release of the highly toxic cyanide involves enzymes, the most important of which is beta-glucosidase, ruminants and llamas and alpacas are most susceptible to poisoning due to the high level of this enzyme within the rumen and compartment of these species. There are 2 main factors that influence the occurrence of cyanide poisoning in livestock. The first is the rate of intake versus detoxification. Hungry animals that consume plants rapidly are most at risk, and hay is more hazardous than fresh plant due to its higher dry matter intake. As mentioned previously, this level of beta-glucosidase in the rumen will influence the susceptibility of an animal to poisoning, as will the animal's ability to detoxify poisonous levels of cyanide based on previous non-toxic exposures. The second is the capacity of plants to produce cyanogenic glycosides. This may depend on genetics. For example, particular cultivars are known to produce higher levels, the stage of growth, the environment, stress, and plant nutrition. Plants with a hydrogen cyanide concentration of greater than 600 parts per million are considered potentially poisonous. Here is an example of the differing levels of cyanide within the sorghum crop, with the most severely drought-affected plants having the most lethal concentrations of cyanide, remembering that anything above 600 is considered hazardous. Animals poisoned by cyanide are usually found dead. However, in those that are seen alive, clinical signs include rapid, laboured breathing, a weak, irregular pulse, and muscle weakness or twitching before animals may stagger and collapse into convulsions and rapid death. Blood and mucous membranes may appear bright red. A tentative diagnosis can be made where there is a syndrome of death and a history of recent exposure to plants known or suspected to contain elevated cyanide levels. The finding of bright red blood may also be useful. However, there are no specific gross or microscopic changes. Confirmation therefore relies upon detection of high levels of cyanide in affected animals. Because cyanide is volatile and rapidly lost from certain sites, it is important to collect samples as soon as possible after death, particularly ocular and rumen fluid, and freeze these samples where possible. Suspected plant samples may also be assayed for their cyanide potential. A cyanide level above 600 can be considered hazardous to livestock. However, levels down to about 200 may be dangerous for very hungry animals. And remember, when sending plant material in for assay, it's important to wrap these in paper, not plastic, and to prevent any damage to fresh material. Moving on to nitrate-nitrite poisoning as another common cause of sudden death. This poisoning usually occurs when stock consume crops, pasture plants or weeds that have accumulated hazardous amounts of nitrate. with numerous plant species known to contain significant nitrate concentrations, including some of our major crop species, like oats. Like with cyanide poisoning, ruminants and alpacas and llamas are susceptible to poisoning because of their ruminal or stomach environment, and in this case they contain microbes that convert plant nitrate into nitrite. The occurrence of poisoning in livestock is a result of incompatible plant and animal factors, where an excess of plant nitrate promotes absorption of toxic nitrite within the animal. Circumstances that lead to a build-up of potentially toxic amounts of nitrite in plants are those that promote nitrate uptake, such as fertiliser or effluent, leading to high soil nitrogen supply, and those that prevent the nitrate conversion to protein, such as low light intensity or plant tissue damage. Ruminants are likely to be poisoned if the intake is rapid and rumen bacteria are unadapted, because the conversion of nitrite to ammonia becomes the rate-limiting step, and toxic amounts of nitrite are produced in the rumen and subsequently absorbed into circulation, converting the hemoglobin in red blood cells from its normal red oxygen-carrying state to the brown non-oxygen-carrying state of met hemoglobin. If more than approximately 75% is converted to met hemoglobin, vital organs such as the brain, heart and liver become starved of oxygen. Poisoned animals are mostly found dead. However, live affected animals may show signs of oxygen deprivation similar to that described for cyanide poisoning. Brown discoloration of mucous membranes may be observed, and pregnant animals are likely to abort. A tentative diagnosis of nitrate-nitrite poisoning can be made where there is a syndrome of sudden death and a history of recent exposure. Animals necropsied soon after death will have chocolate-coloured blood. However, this discoloration fades with time and may not be evident in animals that have been dead for more than just a few hours. There are no other specific gross or microscopic changes. Confirmation is therefore reliant upon detection of high levels of nitrate and/or nitrite in ocular fluid from affected animals. Now remember, because nitrate can be produced by bacterial breakdown of tissues post-mortem, it is important to collect samples as soon as possible after death. It is also preferable to extract aqueous humour into a clean container rather than to submit a whole eyeball. Suspected plant sources can also be assayed for their nitrate content. A level of 1.5% or greater, which is usually expressed as potassium nitrate equivalent, is considered hazardous to livestock. And as you can see here, this plant detected had 7.4%. Another common cause of sudden death is soluble oxalate, which may be present in numerous plant sources. The most commonly encountered in Northern Australia include pigweeds, the grasses Buffalenceteria, Rubidoc, and soft roly-poly. Ruminants are the usual victims of this type of poisoning. Soluble oxalate concentrations are greatest in young, actively growing leaves, and like nitrates, their concentrations can be substantially boosted by soils high in nitrogen from fertilisation or manure, which is readily built up in and around stockyards. Hungry animals that eat large amounts of hazardous plants quickly are the most likely to be poisoned, while animals that have had previous access to below toxic amounts of soluble oxalates will have adaptation of their rumen bacteria, allowing them to safely eat plants that would otherwise be poisonous. Exposure to soluble oxalates removes calcium from the bloodstream as it binds to the oxalate, forming calcium oxalate crystals. The resultant hypocalcemia may result in animals simply being found dead, while others may show typical signs of hypocalcemia, such as difficulty breathing, muscle twitching, staggering, collapse and coma. Crystals may be found in the walls of the rumen, but are usually formed within renal tubules, where they cause renal failure in animals that survive the acute episode of hypocalcemia. Clinical signs and a history of access to plants containing soluble oxalates may lead you to a tentative diagnosis. The presence of hypocalcemia can be confirmed by antemortem biochemistry. However, the histological detection of nephrosis with calcium oxalate crystals in kidney sections is diagnostic for this poisoning. Additionally, specialised laboratories may be able to assay suspect plant sources for their soluble oxalate content, with concentrations greater than 2 to 2.5% indicative of hazardous material. Sudden death is often the sequelae of exposure to toxicants that may have an acute effect on the heart. Commonly encountered myocardial toxins of plant origin include cardiac glycosides, found in plants such as rubber vine, mother of millions and oleander, fluoroacetate in gastrolobium species and Acacia georgina, diterpenoid alkaloids in ironwood, and gossypol from cottonseed. Poisoning circumstances for each of these toxins vary. Plants containing cardiac glycosides are usually not palatable, so are rarely eaten in their normal state unless other feed is very scarce. However, because toxicity is retained in dried plants, feeding of garden waste or contaminated hay is more commonly associated with this type of poisoning. Conversely, plants containing fluorooxalates are quite palatable to herbivores, and cattle, sheep, pigs and horses are all susceptible. Interestingly, native animals that live in habitats with numerous highly toxic gastrolobium species, such as in southwestern Australia, are extremely tolerant compared with the same animal species in other parts of Australia. For example, a brush-tailed possum in Brisbane is approximately 120 times more sensitive to fluoroacetate than a brush-tailed possum in Perth. Cattle and horses are susceptible to ironwood poisoning, and whilst all parts of the trees are considered toxic, poisoning is more often associated with suckers due to their accessibility. Pigs, calves and lambs are approximately 10 times more sensitive to gossypol poisoning than mature ruminants, so cases are most common in these animals or when cottonseed forms a large proportion of the diet, such as when drought feeding. Obviously, the heart is the main organ affected by these compounds. The rate and rhythm of the heart may be affected. Animals may have difficulty breathing and may die suddenly, particularly if handled or exercised. The lungs, gastrointestinal tract, nervous system and sometimes kidneys may be involved either directly or secondarily. Nervous signs predominate in pigs and carnivores, although herbivores may show mild nervous signs. Diarrhea, usually with blood, is commonly seen with cardiac glycosides and sometimes ironwood poisoning. Of note, carcasses of animals poisoned by plants containing fluoroacetate may cause secondary poisoning in scavengers such as dogs, dingoes and pigs. Diagnosis may be suspected where there is a history of access in animals dying suddenly. Gross changes of cardiac muscle pallor and pulmonary edema or atelectasis may strengthen your suspicions of acute heart failure, and sometimes there may be recognisable plant material in the stomach or rumen contents of animals that have died rapidly. Although not specific for the type of myocardial toxin, laboratory confirmation is achieved by the finding of necrosis in histological sections of heart muscle. As the lesions can sometimes be focal, ensure multiple pieces of heart are submitted to the lab. It's also important to include other tissues, including skeletal muscle, particularly in animals that have exposure to monensin, as ionophore toxicity is a significant differential diagnosis for myocardial necrosis. Additionally, rumen contents may be assayed for identification of plant material or fluoroacetate content, or feed may be examined for the presence of contaminant seeds or plants, and gossypol may also be assayed at specialised laboratories.